last from University of Bristol, and he will explain about the implication of aerosol degradation on microbial infections. Thank you very much.
Thank you. Cool. Cool. Thank you. Good morning. I'm here to talk about the implications of aerosol degradation on pathogen detection. Aerosol collection in the environment is relevant to many scientific fields. These include, but aren't limited to, human, animal and plant disease surveillance, studying the aerosol microbiome, and biodefense or pandemic preparedness. In order to be collected from the aerosol phase, bioaerosol has to be first generated, and then pathogen-laden droplets have to travel through the air to that collection point. Whilst this is occurring, there's a whole host of atmospheric, environmental and microbial factors that are imparting stresses on those microorganisms during those three phases of the aerobiological pathway. And each of these factors are likely to have a biological effect on that microorganism. And those can include oxidation and degradation of lipids, proteins, and nucleic acids, which are all targets for detection of pathogens in the environment. There's a lot of literature out there that suggests that these biological effects can impact the viability and infectivity of microorganisms following this process. But what is less well known is whether these biological effects impact our ability to either detect or measure those pathogens within air samples. So just to highlight some of the data that's in the literature, this was a study that was conducted fairly recently, and this group compared a number of different samplers, which both included filter-based collection methods and liquid-based collection methods. And they looked at the aerosol stability and recovery of those. And what they showed that across both those types of technology, both filter and liquid-based collection, they saw long-term air sampling over a period of 2 hours impacted nucleic acid recovery and its stability. So following the collection of aerosols in the environment, there are a number of ways that we can detect pathogens. Some of those are what we call nucleic acid amplification techniques. Those include the polymerase chain reaction and a relatively new technique called loop-mediated isothermal amplification, or LAMP for short. They include field deployable sequencing technologies, such as the Oxford Nanopore MinION platform, but also miniaturized immunoassays, such as the lateral flow device. And for those of you who are less familiar with those, PCR and LAMP basically is detecting a specific part of that bacterial DNA or RNA, and immunoassays are looking at specific proteins on the surface of the microorganism. So very briefly, to describe how physical DNA damage may impact these technologies, PCR and LAMP requires the binding of both a primer, which is a small sequence of DNA complementary to the target template, and a fluorescent probe. And if the DNA at that target region is intact, those primers and probe will bind to that DNA, and with the use of a DNA polymerase enzyme, will extend that DNA. And then with cycles of denaturation and annealing, what you see is an exponential and a sigmoidal increase in the fluorescence, which you can use as a measure of the amount of target that is there. In the instance where DNA damage has occurred, for example, through a single strand or double strand break, these breaks may prevent the binding of either the primer or the probe, or prevent the extension taking place, and therefore prevent amplification. And in those instances, you don't see that amplification, and therefore potentially could see a false negative. So this project in its early phases looked at addressing the question of whether long-term air sampling causes excessive damage to the nucleic acids and whether they affect these technologies such as PCR, LAMP and sequencing. So to test this, aerosols of 2 gram-negative bacteria, Escherichia coli and Pseudomonas batuta, were generated using a 6-jet Collison nebulizer operating at 15 litres a minute for 20 minutes. Those aerosols were mixed with HEPA-filtered air within a linear wind tunnel, and within that wind tunnel, aerosol samples were collected. They were collected using a 55mm filter with a pump operating at 35 litres a minute. So in addition to that 20 minutes, the pump was operating during that 20 minutes, and then for a further 10 minutes, which in this experiment is termed short-term air sampling, or for a further 5 hours, and I've termed that long-term air sampling. Those filters were collected, they were eluted into buffer and shaken. The filter material was then removed using a cell strainer, and that buffer solution was centrifuged for 30 minutes to pellet the bacterial material, and then that pellet was then subjected to DNA extraction using a commercial off-the-shelf DNA extraction kit. That DNA was then analysed using an automated electrophoresis machine at the Agilent tape station, and from that you can get a number of readouts. One of those is a gel image, as you can see on the screen. You can also get what's called an electropherogram, which is basically the size distribution of fragments on the x-axis against their intensity on the y. And the computer software also assigns a score to the DNA referred to as the DNA integrity number, which is based on that degree of smearing that you can see on those capillary electrophoresis traces. So what did we see for both of these species? Short-term and long-term air sampling resulted in a decrease in that integrity score. However, when I viewed the electropherograms, you can see here that there's actually quite minimal damage, and that the majority of the DNA is of high quality size. It's large fragment sizes. With Batuta, Pseudomonas batuta, you can see that there's an ever so slight increase in the intensity of those fragment sizes that are slightly smaller, but the majority of the DNA is of a large fragment size. So I concluded from that DNA degradation was unlikely to impact these nucleic acid amplification techniques because of that amount of a large fragment size of DNA. But that comes with the caveat that all of these experiments were conducted in HEPA-filtered air, and if those bugs were exposed to different environmental stressors, there may be a greater impact on that DNA quality. So really that begged the question, how much damage is needed to impact these technologies? And in this instance, cultures of E. coli were grown overnight, DNA was extracted from those, and that DNA was then subjected to various times of sonication. Sonication is well known to cause physical damage to DNA and is used in whole genome sequencing. Those fragment sizes were then analysed by the tape station instrument, and then were either run on a Quant 7 PCR instrument or a Genie 3 LAMP instrument. And what we saw was actually that sonication works really well as a positive control for DNA damage. You can see in the left graph there, when DNA was not sonicated, an extremely large portion of that DNA is of very high quality. Those fragment sizes are very large, and on this instance, with a mean peak base pair size of over 58,000. But even with just 40 seconds of sonication, you can see that size distribution moves quite far to the left, and in this instance, with a mean peak base pair fragment size of just under 500 base pairs in size. If you increase that up to 12 minutes, that then decreases to below 200 base pairs. So what impact does this have on PCR and LAMP? For PCR, not much. Even at those large sonication times, there's not much of a difference in that crossing threshold value. Starting to creep up at that 12-minute mark, but that's minimal. For LAMP, even though the results were non-significant, you can see there that there's a definite trend that with increasing sonication time, there's an increase in amplification time. So to conclude from that, LAMP seems more susceptible to DNA damage, and it's hypothesized that that is as a result of the larger amplicon size associated with LAMP. So the amplicon is the proportion of DNA that is amplified during that process. And that PCR, which is typically has a smaller amplicon size, may be more resistant to that damage. So the next question to address was really, did PCR, was the limit of detection of PCR impacted by this damage? So in these instances, following extraction, DNA was sonicated, run on the tape station, and then serially diluted between neats and -10, and those dilutions were then run on the PCR instrument. And you can see from the graph on the right, even with sonication time of 12 minutes, those traces are almost identical to the control. Just to address the question of whether the concentration had an effect and whether it was more realistic to real life, we changed the method ever so slightly, diluted the samples, sonicated them, ran them on the PCR instrument, and again, there was very little difference. So to conclude, long-term air sampling decreased DNA integrity, but it's unlikely to impact these technologies. LAMP was more susceptible to DNA compared to PCR, and this physical damage did not affect the limit of detection. Very quickly, the future work will look at comparing PCR to LAMP. The effect of physical damage and long-term air sampling will be assessed with third-generation sequencing technologies. I'll look at the antigenic structure, and further studies will look at the effect of environmental conditions on these assays using the QLabs and the BioArc, which is at the University of Nebraska. Thank you.
Are there any questions?
Thank you. I mean, interesting talk. If I look at the recent experiments, was the size like tens of thousands? Is that base pairs? Is that what you were seeing?
Yes.
It's obviously an eco genome about a few million, right? I mean, but obviously you need to extract the DNA from the cell. So I mean, if you just took like E. coli, without putting an aerosol, and you extracted it, you also get fragments of that size. I guess I'm saying, is fragmentation caused by the aerosolization or caused by obviously the fact that you extract it from the cell anyway?
Yeah, it's a good question. I guess it's unknown because there are a whole host of processes that you need to do. So is that occurring at the aerosolization stage? Is it occurring at the centrifugation stage, for example? So to extract and dissect all of those components is probably a PhD in itself. But
yeah, it might worth comparing. I mean, obviously, the infectious in time between kind of which obviously inside the cell, then hopefully. Okay, thank you.
thank you.
Question?
Say that Cohort here, it's a really nice one. Just have one question. Is there anything upstream of centrifugation or nucleic extraction that you would do to prevent further DNA damage done?
So only other than the kind of typical approaches, so keeping DNA on ice, for example, using nuclease-free water, just to prevent those enzymatic reactions that could degrade DNA in those samples.
There's nothing in the sampling section of your process that you would do to prevent?
Not currently.
Thank you for the nice talk. Everybody from the University Foundation. I was wondering, were you using the same targets when you were comparing the breakage retention of DNA between the lab and the PCR?
No, they are different targets.
The different targets.
Thank you very much. We can move to the next one.